I will be talking the photophysical dynamics or like the cluster. So here this work we have done in collaboration with Professor Patra at INST. So here we have used two nanocluster, two types of nanocluster. One is silver 29, another silver 28. One silver it was replaced with one gold. So in the morning Professor Goswami has Professor Pathak already told that after like changing or replacing one atom, it can significantly change the activity of the nanocluster. So similarly here also we have studied that how the dynamics is changing after doping with one gold atom. So these are the like the fluorescence spectrum of this kind of gold and gold silver. So we have found that after doping the fluorescent intensity it was increased, and this nanostructure it was stabilized with DHLA, this hydrolipoic acid. So these are the TM images, and the size of this structure is around 1.9 nanometer. So after that we have used confocal fluorescence confocal microscope, and we have studied this system. And here this is the like the fluorescence lifetime images of this without silver, with only silver, and this is with gold after replacing one atom with gold. So here you can see that the intensity of the spot it is much higher after doping with gold. So the intensity it was increasing after doping, as well as we found that here is undergoing bleaching after 20 seconds, but here is giving the emission till 100 seconds. So after gold doping, not only the intensity is increasing, but also it's improving the photostability. So we have characterized the photo blinking dynamics, and for like few nanoclusters we have like carried out we have accumulated data from at least 100 nanostars nanoclusters, and here we have found that from some nanoclusters this beautiful blinking is coming. So we have also tried to understand that how much is in which time scale the blinking is coming, and what is the time scale of the on off time also. Here you can see that signal intensity is very high, and also we carried out here is showing single step bleaching. Yesterday Professor Nondi already told this is the signature of single nanocluster present in the confocal region. So in our case also mostly we have got single step bleaching or single step blinking. So it was confirming, and in addition to that we have carried out the anti-bunching like measurement. It was also confirming that in the focal volume only single nanocluster is present. So we have further like characterized, as I told, that at least 100 structures we have characterized, and we have accumulated data, and from that we have got the statistics, and it was found that in case of the like nanostar here in the pure silver nanoclusters, this life is around 50 is around 80 millisecond, and here is the off time, this is after replacement of one atom is around 10 millisecond. So this is the on time and off time for silver, pure silver nanocluster, and this is the on time and off time of the doped nanostar. So it was indicating that all the time scale is in the millisecond range. So generally in single molecule, if we carry out the measurement with fluorophore, then this blinking is very fast, and the photo bleaching is also very fast. So generally people use some oxygen scavenger and also Trolox kind of thing, so that it can increase the stability as well as it can decrease the photo blinking events. So in our case, without using any such oxygen scavenger, we found that the on time is very high. You can use this as excellent probe to carry out the single bimolecular measurement, and also the off time, this is also very high. That means you will have very less blinking in the time scale of your measurement. So these are the like again photo stability I was talking about. After replacement of one atom with gold, you can see this is the photo bleaching. It undergoes photo bleaching after 20 seconds, and here it was stable almost till 300 seconds. So here the blinking we have found, but in case of the silver nanocluster we have found very less blinking. So we have calculated the survival time. We have checked the survival time of gold pure silver nanocluster as well as doped silver nanocluster, and we found that here it was giving the emission till 65. After that it was completely bleached. But in case of this doped system, we found that it was giving this cycle, this blinking cycle is stable till 280 18 seconds, and after that it was undergoing bleaching. So that means this will be much better probe compared to the silver nanocluster. And we have calculated the range, like this is a range of the survival time, and in this in the case of the pure silver nanoclusters, this range is around 20 to 30 seconds here, and here this range is around 200 seconds. So significantly the photo stability is increasing after doping with only single gold atom. So we have like, this is the intensity, this is the like from the, as I told that many structures we have taken into consideration, and after that we found that approximate 5-fold enhancement in the intensity we found after when it was doped with single gold. And the photo stability, it was also in case of the dope structures we found longer on time and shorter off time, and it was more stable. So this kind of system can be used as excellent fluorescent probes for single bimolecular measurement in both shorter time scale and longer time scale. So this is very interesting finding. So far in literature we have found that some reports on single molecule nanoclusters were there using the DNA as the stabilizer, but in those cases people have reported that the blinking or the bleaching is much faster and the blinking in the range of microsecond. But here we have shown that the blinking or the one time is in the millisecond. So this is much higher. So, and this is very good for single molecule measurement. So after that, like we also, as I told, DNA origami, we wanted to play with the like the stoichiometry, as well as we wanted to play with the material, and we were able to put or assemble some kind of quantum dots, as well as we were interested to create some kind of DNA origami structure which contains single nanocluster. So we have like used this, we have this DNA origami we have modified with polycytosine, and we have used this HUCl4, and in the with the reduction of DMAB we found that this DNA origami template in situ synthesis of gold nanocluster taken place. So here these are the AFM images and TM images. Here the AFM images, it was indicating that gold nanostar is present on top of the DNA origami. So this is the height profile, this 2 nanometer height, this is the standard double helix structure, this is due to the DNA origami, and this further increase of around 2 nanometer, this is due to the immobilization of the nanocluster on top of the origami. So it was indicating the size of the nanocluster in our system, this is less than 2 nanometer. So these are the TM images, and here you can see that we could not go further higher resolution, because if we use more like here magnetic field, then our DNA origami, it will be destroyed. So here we have carried out this line EDAX, and it was giving the signal of gold. So it was further indicating that gold nanostructure is present on top of the DNA origami. So this structure also we will further use it to have the high resolution and to get the size of the nanoclusters. So we have like again carried out similar kind of measurement, and this kind of cluster also interestingly we found it's also showing the blinking. So it's not only depending on the ligand, because here the ligand is different. So here also we have found that the on time is also very high here, and the lifetime in the range of nanosecond. We did not see any microsecond component here also. So these are the again film images, and we have found some blinking events here. So we are these are the very preliminary data. We are trying to have more statistics, and we are trying to calculate what is the on time and off time for this kind of nanocluster on DNA origami. So I want to thank my students. So the JAX paper and the initial data I have shown Swati's work here. She is presently working as the postdoctoral fellow in Johns Hopkins University. My second student also joined the same group, and Vishaldeep recently left my group, and he joined as the postdoctoral fellow in Indo-France University, Netherlands. She carried out of the measurement where I showed the gold nanocluster and silver nanocluster here. So I am thankful to my recent student Charanlin, Mridu, Shikha, Priyanka for their significant contribution in my lab. I am thankful to my collaborators, Dr. Shungita Saini from Kurukshetra University. She helped us in the electric field calculation of the different nanostructures, and Professor Amtav Patro, who is the director, I am thankful for his immense support. And I am thankful to my collaborator Ashif Khan, who is present here. He helped us in the photothermal therapy, that kind of application by using this kind of nanostar application. Dr. Krishnakanto Haldar. We do collaboration for measurement, some kind of electrochemical measurement. And of course, thanks to the funding DST, ECR grant and CRG, with the help of this grant, I was able to set up this single molecule confocal. And of course, with the help of INST, we were able to successfully establish this single molecule confocal facility at INST. Thank you very much for your kind attention.
Thank you very much, Tapasi. I'm sure there will be questions.
Thank you very much. I have a question regarding your system with the nanostars. So I saw from the TM you have spikes of different lengths, and I know that if you conjugate to the tips or just on the edge, you can have a fall of more than 3 orders of the cells. So how do you manage to have a good reproducibility of your measurement looking at the polydispersity of the spikes on your stars?
So here this kind of structure?
No, I'm speaking about the stars.
The stars one.
The gold
so
nanostars.
Stars one, actually, this is very important because it's really very difficult to control over the orientation. So some of the structure we have found is giving very strong enhancement of the Raman signal, as well as some spots we have found or some structures we have found, those are not giving enhancement. Even sometimes we have found is giving the quenching also. So it's really very difficult because sometimes due to the structure the tips are touching, and we have found from our previous like report in the JAX we have shown there, when the tips will be touching, there will be in addition like instead of the like electromagnetic field enhancement, there will be drastic quenching. So this is really it's very difficult. That's why presently we have moved to the other system, the bipyramid one, because this is here the orientation is much better. You can control the orientation in much better well compared to the nanostar. But of course, nanostar are it can give much more significant enhancement compared to because they are having multiple tips. Okay. But the control there will be much less compared to the bipyramid.
Okay, thank you.
There's a question back in the back there.
Ma'am, can you explain the sensitivity of your substrate?
Sorry,
sensitivity.
Sensitivity?
Yes.
I have told the sensitivity is single molecule level. In most of the system we have found single molecule. So it will be, if you consider in the concentration, femtomolar.
Okay, thank you, ma'am.
Very good presentation, Dr. Tapasi. Here.
Thank you, ma'am.
Okay, so I have few doubts. One is that golden nano star, where are you embedding the star in the origami template, or are you conjugating the origami to the tips of the star?
First we have prepared. So for example, this is the origami, right? And we have used some capturing stains which will be like here in the upper direction it will be there. Now we have we have prepared separately gold nanostar which are functionalized with DNA, single stranded DNA. Now this capturing sequence and the capturing sequence which are used to functionalize the nanostar, both are complementary. So the moment you will be mixed, it will be sitting on top of the origami. Okay. Specifically, it will be sitting.
So then there is another doubt. What is the significance of origami? Even a single stranded DNA should work, no?
You cannot play with the gap. As I told that with single stranded DNA is forming and finally is making the double stranded DNA with the help of this base pairing.
Only one more doubt. So you are telling that fluorescence is enhancing and the Raman signal is also enhancing.
Yes.
So this is somewhat contradicting now. When the fluorescence is enhancing,
it depends on the distance. As I told, this is very sensitive to the distance. So in case of the plasmonic, like this kind of dimer structure, in the hotspot if you place, the fluorescence it will be enhanced as well as the Raman signal. But if you take the single particle, if you just put on the surface, just there is no gap between the fluorophore and the star, and then there will be significantly fluorescence quenching, but you can have Raman enhancement. So it's very deep, it's like the optimum distance is really required to achieve very good enhancement.
Okay, thank you.
Thank you for a very nice talk. Coming back to the sensitivity issue, you mentioned that you detect a single molecule. I understand that. The question when you go to detection is it's going to be diffusion limited. Do you have any idea of the time it would take for an analyte in the solution to come in the right place to be detected?
Because we have not carried out any measurement in the solution phase. In case of the Raman also, we have dropcasted the sample and it's fixed on the surface. so there is no movement. And fluorescence also we have used the immobilization based techniques, and we have not done any kind of FCS. So everything is fixed on the surface. So
Yes,
there
yes,
is no
but-
diffusion is happening.
Yeah, I understand in your experiments there is no diffusion,
Yes.
but in real life, if you want to use this as a sensor, you would have to catch that very
So in
lonely
case of
molecule in the solution.
yes, we understand that. But maybe, some people have used this kind of nano antenna to understand the holiday junction kind of thing. So this kind of diffusion you can play there. It has not necessarily to be attached on the surface, but still you can use. The moment it will be coming close to it, you will have signal enhancement. When it will go far apart, then the signal it will be reduced.
Martin.
Okay, thank you very much. This is really impressive work. I have many questions, but I think I'll look up the papers and I'll find the details there. I was wondering, when you make the dimers, do you also measure the broadband light scattering spectrum, like the dark field scattering spectrum?
That we have not done. We plan to use it, but here we did not have that setup here in INST. But now we are having this setup, and we can plan for that. We also have in our mind how the LSPR also changes, and with the interaction with two plasmons. Yeah. And dark field also, we will have different colors also.
And one small technical question.
Yes.
For the excitation of the single nanocluster objects, when you compare that to the excitation of single molecules like cyanine dyes or fluorescents, what is the relative laser excitation intensity or fluence that you
Laser we use
need?
in the range of microwatt.
For both
the
of them?
Psi3 we have used 0.1 microwatt, something like that. And for the nanocluster we have used 1.4 microwatt, like that.
Okay.
But it's in the range of microwatt because we did not want to have this photo quenching.
Okay, thank you.
Photo bleaching we did not want.
Okay, last question.
Yes.
I mean, this is a very interesting lecture, first of all. So I was wondering the fact that when you have one gold atom in a silver matrix of 28 silver atoms, why is there an enhancement? So what does the silver do? Does it funnel the energy to the gold?
That is really, we don't
That's
know
a
yet.
very,
More
very
fundamental understanding
Yes,
is
I was
needed.
trying to think.
Yeah, because this study, this is the
I
whatever.
was trying to think, trying to connect it to kind of those photosynthetic proteins, the energy funnel-like model.
But here nothing like
There
that.
is no harvesting
no,
like
no, no harvesting here.
Okay.
So, yeah.
Yeah, please.
That time kernel had changed. So bimetallic kernel, their photoorbital will be changed. So two metal are given, now that system is changed. That is reported structure.
Yes,
then
so
we can have
here upon introduction of gold, some new bond will be formed. There initially there was only silver. But still we have to study the system in more detail to understand the what is the mechanism of the blinking and all other the photo stability.
The band gap of 28 of them must respect the scale with respect to number of atoms. Maybe we can discuss.
Okay. Yeah, we should keep that short because at some point we have to be able to eat all the things that are there. I saw there is another last question.
Just a comment, not question. Question we have later, several we can discuss later on. So I saw that your single molecules time traces, there are a lot of spikes. So I think that you need
This
to
is
take
coming
care.
due to the nanostructures.
No, not really. That may be something different. You should look into it because I saw a lot of background noises as well. While the single step bleaching already happened, still you have a lot of background noise.
So generally.
So those are the noises, I think, somehow.
Yeah, so in case of the nanocluster, there might be some aggregation and scattering comes, but in case of the the nanostars or the dimer antenna, so already people have reported that due to the scattering of because some nanostars are there, so some background already people have reported.
Okay.
So if you see Phillips' paper, then already they reported due to the scattering coming from the big particles.
Okay.
Okay, I think with this we should finish the discussion. Thank you very much.
Long live India and France friendship.